infection over Treponema pallidum. Here are some of its characteristics. It has a twisted corkscrew shape. It is a gram-negative bacterium, and it has a cell wall with a sufficient amount of peptoglycan in it. It also contains the flagella. Here are some odd facts. On this side you see T. pallidum, and on this side you see a standard gram-negative bacterium. T. pallidum. The peptoglycan lays over the cytoplasmic membrane, but on a standard bacteria, the peptoglycan lays underneath the cytoplasmic membrane. For T. pallidum, it has a relatively high amount of lipoproteins, but on a standard bacteria, it has little to no lipoproteins at all. It also has a fascinating flagella, typically 3 flagella, strategically placed around the bacterium. This helps it have a free range of motility. It is able to flee or attack. If something is trying to invade it, it can flee away from it, or if it wants to attack something, it can attack towards it. This flagella is made up of durable proteins. This helps it travel through highly viscous conditions, such as connective tissue, so it can literally corkscrew its way into your skin. Here are some striking characteristics. It has a shortage of tricarbonic acid enzymes. These enzymes help speed up metabolism. For its metabolism process, it has a glycolysis process, which is an anaerobic process, which leaves it with 2 ATPs. Here we see E. coli, a gram-negative bacteria that uses an aerobic process. This gives it 38 ATPs every 20 minutes. This is 19 times more than T. pallidum. Perhaps this is why T. pallidum has to use its host macromolecules to help make up for the lack of ATP it makes. There is very little known about the T. pallidum bacterium because it has not successfully been grown in vitro. Also, it is only able to live in a human mammal host. However, studies have shown that it has lived longer in vitro when anaerobic processes are performed. This led scientists to believe that it is anaerobic. However, recent studies have shown that it is not. It is microaerophilic, which means T. pallidum does need oxygen to respire, but too much can poison them, so it lives in a low concentration of oxygen areas. Its attachment site. T. pallidum is a sexually transmitted disease. Its transmission is when it comes into contact with moist openings, usually, but it can penetrate the epithelium. Once it is exposed in a human body, it is able to travel in the blood. From there, it can attack numerous cell types. As I said before, T. pallidum causes syphilis. After 3 weeks of exposure, it reaches the primary stage. These include a painless, clear, ulcer-like lesion. After 6 weeks, this lesion is cured. Next, it goes into secondary stage, which is usually 3 months after exposure. Some symptoms include muscle soreness, discomfort, and/or sore throat. Other signs of second-stage syphilis are cutaneous rash found on the palms of the hand or the soles of the feet, enlarged ulcers on damp, moist areas, and/or non-tender lesions that can sometimes become painful. When it's not in primary stage syphilis or secondary stage syphilis, it is said to be in the latent stage. This is where the T. pallidum bacteria remains dormant in the body, but there are no symptoms for it. Lastly is the tertiary stage. This is a life-threatening stage when the T. pallidum bacteria reaches the cardiovascular system or the nervous system. However, this stage is very rare because antibiotic therapy has been successful. There is also congenital syphilis. This is when a woman has syphilis and is pregnant with a baby. The The T. pallidum bacteria can pass through the placenta. So if left untreated, the infant can either die, become damaged, and or be born with infected and have all these signs and symptoms. Scientists have still been looking for a cure. In the 1970s, heat therapy was considered. Studies have shown that at least one T. pallidum dies at human body temperature, so it has a limited heat range. A Vietnamese doctor decided he would raise the temperature of his patients and see if it would kill the bacteria. It did successfully kill the bacteria, however, a high percentage of his patients died as well. Another cure was penicillin. The peptoglycan on the outer cell wall is very sensitive to penicillin. Penicillin blocks the binding sites of T. pallidum and other cells, so T. pallidum is not able to attach itself to cell types. Therefore, this has been successfully tested to terminate T. pallidum production with the help of the immune system. However, it is not 100% cure because it does not work in every patient. Here are some other treatments. Many campaigns using lotions, creams, and washing your hands and body thoroughly can decrease the production of T. pallidum. However, studies have shown that it is highly ineffective. Another treatment could be condoms, but it is not 100% safe either, because syphilis is very contagious and kissing can expose an individual to the bacteria as well. As we said earlier, heat therapy was unsuccessful. So lastly, antibiotic therapy is the only successful treatment as of today. In conclusion, T. pallidum has a wide range of motility. This makes this bacterium very dangerous because it can affect and attach itself to any cell type possible. After exposure, syphilis comes into play, which, as you saw before, the signs and symptoms are very painful. However, there are treatments such as penicillin that can keep the patient from reaching tertiary stage syphilis. So, if you or an individual is infected with syphilis, you should see your doctor immediately.